All right, that's a log phase of growth. It's also called the exponential phase of growth. So because it grows pretty rapidly. So this is this one. Grows pretty rapidly, and then at some point right here, you run out of nutrients. So there's less nutrients in the environment. Well, bacteria not grow so rapidly anymore. In fact, the best way to characterize this phase is called a stationary phase. There's death equals to life. So basically, the number of new bacteria that's created is kind of generally equal to the number that die. So this is stabilized, right? So we have a lag phase. Nothing's happening. The bacteria is getting ready to use the nutrients, but when it has all those enzymes ready to go, it replicates itself pretty rapidly. I now enter the logarithmic phase. It's also called the exponential phase of growth. By the way, this is the best time to kill bacteria is when they're growing pretty rapidly. It's the best time to use antibiotics to kill bacterial cells. And we're going to talk about that later on in chapter 10. At some point, you run out of nutrients. So basically, this actually subsides. Death equals to life. There's no new bacteria net speaking. So basically, the net is zero. And then there's no nutrients back here. Bacteria start dying. This is called the decline phase or the death phase. So we have the lag, the log, we have the stationary, and then we have the decline or death phase. Now, the next thing I want to address is this. Imagine this is E. coli or Staphylococcus aureus. Once you get to this point back here, basically, your cells are all dead. Basically, they just ceased existing. But some organisms that cause disease in humans, they're pretty fancy, actually. But here's what they can do, okay? They say, you know what? I've been growing pretty nicely in this medium, but now I'm running out of nutrients. And if I don't do something, basically I'm totally obliterated from life. Well, I want you to remember this. There are two groups of bacteria that do something very special. These bacteria are Bacillus and Clostridium. If you remember, Bacillus, they're aerobic rods. So we talked about the Gram stains way back when they're gram-positive rods, but they like oxygen. The other one, and for Bacillus anthracis, this is the one that causes anthrax. The other one is Clostridium. So this is the other group here. Clostridium are anaerobic gram-positive rods. So think about this. This one doesn't like to be around oxygen. It's anaerobic. Let me give you an example. Tetanus is caused by Clostridium tetani. So that's an anaerobic organism. Anthrax is caused by Bacillus anthracis. That's an aerobic organism. But let's get back to what we're talking about here. In the case I showed you earlier, once E. coli runs out of nutrients, basically it would die. It would cease to exist. These two groups of bacteria, Bacillus and Clostridium, they do something pretty cool. They kind of detect, you know what, nutrients running out. And if I just keep on growing, or I keep on just maintaining my existence there, as is, I'm going to die. I'm going to be erased from life. All right? So here's what they do. They form spores. Now, way to think about spores, think about seeds, plant seeds. You can take corn, for example, get the seeds and store it for years. And then you plant it again, it germinates, and then you have a new plant. Well, these bacteria are thinking basically the same thing. You know, here's what they're saying. Let me go back to this slide. They get to this level here. This is the stationary phase. They were already thinking biochemically, you know what? If I continue, I'm not going to exist anymore. I'm going to start doing something really cool. I'm going to start forming spores. Now, these are the two major groups of bacteria that cause disease in humans that can do this. The others can't. They can form spores, all right? So here's what they're thinking. All right, let's start at this point. They're replicating their DNA. Nutrients start running out. They detect this, and then they form a spore. So basically, this is not a mature spore yet, right? They already know nutrients are going to run out. I better start doing this process. Pretty much this spore develops into what's called an endospore. So this is the endospore. Endo means inside. It's inside the mother cell. And then what happens, pretty soon after that, enzymes are produced to destroy the mother cell here. So this doesn't exist anymore. That spore, that endospore, is now released. This is called the free spore, or it's called a spore. So let's talk about those two diseases I mentioned earlier. Anthrax, caused by Bacillus anthracis, and then tetanus, caused by Clostridium tetani. Well, how do we get those diseases typically? So here's what happens. People are walking around in the soil, and the spores are present in the soil, and maybe they have a wound on the leg or something. Some of that soil spills into the wound, and that spore germinates, just like a plant seed germinates to create a new plant. And then what's going to happen? That spore germinates to produce a vegetative cell. That's important, all right? This is the cell, that vegetative cell that produces toxins. So when you have a problem, say with tetanus, that toxin that's produced, it's produced by this vegetative cell, not the spore. The spore is dormant. The spore doesn't cause disease. The spore gives rise through a process of germination to the vegetative cell. It's the vegetative cell that's causing the problem, right? This is the cell, the vegetative cell, that produces the toxin. So real fast again, these two groups of organisms, Bacillus and Clostridium, they can cause disease in humans, unlike E. coli and the others. Basically, when they run out of nutrients, they produce spores. These spores can hang around for a long time. Do you know the oldest spores that we know of, this dates back to about 703, not years ago or days ago, 703 million years ago. Scientists found these little salt grains that were dated to that time. They cracked the salt grains open, and guess what? There were spores in there. Those spores grew after 700 million. That tells you something. These spores are resilient. In fact, they're the most resilient or hardy of all the biological entities that we know of. So bacterial spores, they're phenomenal. Just two groups that cause disease in humans that actually can sporulate. That's Clostridium and Bacillus. Clostridium is anaerobic, Bacillus is aerobic, right? Now again, I'm going to point out just a few other characteristics I want you to know about spores, and I can take all of this stuff here and dump it on this slide. By the way, before I get to that slide, these are cells, these are the vegetative cells back here. They're growing. Right over here, this is a forespore or an endospore, all right? And what happens, the mother cell degrades and they release the free spore here. That's what we're looking at. That's the free spore. Very resilient structures. I don't need you to memorize all the specific structures that's indicated here, but just to know these little facts. Spores are dormant. They're extremely stable in the environment. Some of them can withstand high temperatures, 150 degrees centigrade. Think about this. Water boils at 100 degrees centigrade. Some spores can actually survive in boiling water for a long period of time, right? They're resistant to desiccation. Desiccation means drying. They're resistant to acid, bases, chemicals, ultraviolet light, and they can withstand high pressures. So again, very resilient structures. Let me just say one more little note about spores. Most spores, in fact, practically all of them, when they're produced, they have very little water in them. So there's something that replaces water. That chemical is called dipicolinic acid. So again, dipicolinic acid you find in bacterial spores. It composes about 10%. This is 1/10 of the weight of this cell. That tells you it's very important. So basically, it kind of stabilizes the environment when water is not present there. All right, so spores are very resilient. Plus, don't forget it, Clostridium and Bacillus, two groups of bacteria that cause disease in humans that are able to form spores. Spores are dormant. So what happens, you find them primarily in the soil. That's a reservoir for spores. If a spore gets into a wound, it germinates just like a plant seed. And what's produced is a vegetative cell. It's the vegetative cell that's causing the problem because that's the cell that produces the toxins, enzymes that have negative consequences. The spore doesn't do that. All right? This is a picture of a spore or spores germinating, like you see right over here. All right? And again, the spores germinate into something called a vegetative cell. All right, so I want to go back to some of those definitions we talked about earlier. Temperature requirements for growth. Some organisms are psychrophilic. That means they can grow in the cold, even in refrigeration, all right? Mesophilic, that's middle. So middle temperatures. Your body temperature is 37 degrees. These organisms can grow from 10 to 45 degrees fairly well. So most pathogens are mesophilic. And most pathogens here are mesophilic. Thermophiles and hypothermophiles, if you remember, they grow at high temperatures. Generally, they don't cause disease in humans. So psychrophiles can cause disease. Mesophiles can cause disease. Psychrophiles, remember, they can grow in the cold. Mesophiles can go to a range of temperature, well, within your body temperature, which is 37. Here's a nice little chart to show you this. Psychrophile, low temperatures right here. Mesophiles, this red one here, can go in a white temperature range. These other ones, hypothermophiles and thermophiles, they do not cause disease. Why? The temperature for their growth is too high. Like your body temperature is 37. Sometimes a hypothermophile requires 85 degrees to even centigrade to start growing. So again, your body temperature is not even close to that. Right? This is called a concept map. General way of viewing what you've learned. So I don't discuss this here. If you take all those terms we talked about before, halophiles, you guys studied for your quiz today. We talked about extremophiles or hypothermophiles, mesophiles, barophiles. Basically, you can place this in this concept map. All right. What about oxygen requirements? So what we just talked about before, it's temperature requirement. What about oxygen requirements? Some prokaryotes are obligate aerobes. In other words, they absolutely require oxygen to grow. Some are aerotolerant. Some are anaerobic. Oxygen can kill them. Others are facultative. If you remember this term back here, so facultative simply means this. Organisms can grow both in oxygen and without oxygen. So most pathogens are they're mesophilic, but they're also facultative. And capnophilic. Capno refers to carbon dioxide. Phil means to love. So these organisms can grow in fact, they require carbon dioxide to grow. Right? Okay, so this is a slide to show you just some differences between organisms. Of course, it's a little cartoon, it's a drawing, but it exemplifies something very important: anaerobic versus aerobic versus microaerophilic. Now, this is a little tube with broth in there, all right? And bacteria is growing. So in this case, you see bacteria. These little dots represent bacteria. It's growing all over the place. Well, at the very top of the tube, that's where oxygen is present. Here's the air. So air has oxygen. All right, so right in this interface between the air and the medium, there's oxygen there. I have growth. But I also have growth on the bottom part of the tube where there's a lack of oxygen. So this tells you this organism can use oxygen. It can grow with or without oxygen. That's a facultative organism. Right over here. That's facultative. The other one is this. Take a look at this one. It's only growing where oxygen is. So that's an aerobic organism. And then finally, take a look at this one. These are the three major ones. This one's going on the bottom of the tube where there's an absence of oxygen. So that's an anaerobic organism. So don't ever think bacteria, they all go similar and they all love the same type of nutritional requirements or temperature requirements. Not at all. Clostridium, for example, can't do this. It wouldn't grow like this. But it would grow like this here. It would grow in the bottom of the tube. Why? Because it's an anaerobic organism. All right? Okay, so acidophiles, we talked about these terms earlier. All right? And we're done with this lecture. Now, notice there's a section here on media. I'm not going to say much about media. Media is something that you use to grow bacteria on. We're going to talk about this in the laboratory. So we're done with this section here. Right? Okay, do you guys have any questions on this before I attack chapter 20?
Can you mention the things you said again about cholinic acid?
Yeah, okay, so dipicholinic acid. Let me go back to that slide. All right, oops. Dipicholinic acid is a molecule you find in very high concentrations in bacterial spores. You guys see the slides? All right, so right here, this is the chemical. And again, don't memorize the chemical. I'm not going to ask you to draw it. But this chemical you find in very high concentrations in spores. You don't tend to find this chemical in very high concentrations in the vegetative cell. So here's the reason why. When the spore forms, you kick out water. So you have very little water in the spore itself because water can serve to degrade things over time. So you replace water with dipicholinic acid. It keeps all the molecules in there fairly stable. All right?
I have one question, actually.
Yeah, go for it.
So if you were to have cells that are in the decline phase in your body, would the endospores move throughout your body to try and reproduce?
Yes.
Okay.
That's a very good question. So I want you to think about this. Imagine somebody has a gunshot wound. It's a terrible thing to think about. And just imagine I have, let's just say, Clostridium tetani, right? It's in that bullet that enters the wound, that enters the skin. Now that spore is in the wound. So initially, not much is happening because it's not a vegetative cell. But because that wound is going to provide nutrients for that spore will germinate. And of course, when it germinates, I have a vegetative cell that's going to produce that toxin that causes tetanus. That toxin is called tetanospasmin. We're going to learn about tetanus later on, but you're right. But imagine in that wound now, The bacteria uses up that nutrient